You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoy the podcast, please listen, subscribe, like, and share it with friends. Thank you.
Hello, this is Richard Jacobs with the Future Tech and Finding Genius podcast. I have Ivan Poon. He's a senior research fellow in biochemistry and genetics at La Trobe University. We're going to be talking about extracellular vesicles and what happens as part of cell turnover and other things in the body. So, Ivan, thanks for coming.
Yeah, thanks for inviting.
Yeah. Tell me a little bit about your research. What's the focus of it?
Yeah, so the focus of our work is trying to understand what happens to dying cells, when they die. In particular, the process of when they die, they will actually undergo a fragmentation process where they will generate small vesicles called apoptotic bodies. And what we're interested in is, why when cells die, they will spend so much energy to actually generate these vesicles, and the reason for this process, whether it plays a role in facilitating communication with other healthy cells nearby, or is it a process to help neighboring cells to actually clean them up?
So, yeah, I've heard of apoptotic bodies, and I guess they're classified as a type of EV, an extracellular vesicle.
Yep.
I know there's also exosomes and things like that. So what happens visually, and then in other methods, what happens when a cell dies? Does it seem to break apart in an orderly fashion, or does it just explode and fragments of it are all over the place?
Well, I think for many years, at least for the last 40 years, since the discovery of description of apoptosis, people thought that when cells die, they can actually generate a variety of different vesicles, in particular apoptotic bodies. And it's thought to be more of a random process where they just randomly break apart into different fragments, because they thought that it's just a waste disposal kind of process. But more recently, we discovered that when cells do die, it seems to be a highly regulated process. Morphologically, when you look at it under the microscope, they can form a variety of very structured protrusions where they control how cells actually disassemble. And then to support that this is a regulated process, we found a number of molecular regulators that control this process. So recently, we found this process more regulated than random.
Okay, but again, what does it look like? Is it does the cell lyse open, or does it the membrane vesiculate and these gigantic apoptotic bodies spin off, and how many of them come off approximately, millions or thousands, or
One key thing to keep in mind is I think every cell type is a bit different. So for example, for T cells, for example, where when they die, they will actually form circular bulges on the surface first, which we call blebs. And then, the T cells can then form a very thin protrusion, like a string protrusion that helps to separate them. So the cells actually doesn't lyse, but actually form vesicles that is still intact. And in this case, possibly one T cells can generate maybe 10 vesicles that are intact. You can actually see them. They're about 1 to 5 micron in size. But for example, for other cell types like monocytes, what they can do is that they can form, again, when they die, they form this string-like protrusion. But this string-like protrusion that becomes like a beaded necklace. So basically the protrusion first form it becomes beaded. And it's like the fragmentation of this protrusion then generate the apoptotic bodies. In this case, the vesicles or apoptotic bodies are smaller, so they're usually between 1 to 2, 3 microns. But what's interesting is that using this separate mechanism of generating the apoptotic bodies, the cells can actually generate up to 60, even more apoptotic bodies. So it is all cell type dependent.
So a protrusion comes out of the cell and then it beads along the length of the protrusion, and then what happens? Each bead is an apoptotic body, or does this whole string come out, or what happens?
Based on microscopy analysis, we would think that both could happen actually. So because it becomes beaded, so depending on how it fragments, so each of the single beads could be apoptotic bodies, and sometimes they can also attach like a string of beads as well.
Huh. What about looking inside the apoptotic bodies? What's in them?
So a number of work done by others and us have shown that they have a variety of molecules very similar to what's in exosomes and microvesicles. So they can contain DNA, RNA, a variety of proteins, and obviously lipid. But the thing about apoptotic bodies is we still don't really know why some of those molecules are packaged into apoptotic bodies and how they actually function.
So, okay, but I've seen like with exosomes and other EVs, they'll have, various RNAs, they'll have all kinds of stuff. But again, is there a predominance of what is inside an apoptotic body? Has anyone looked? Can they be, can you centrifuge down without destroying everything to look at their structure? Can their structure be seen on microscopes? I mean, what do we know about them?
Yep. So we can actually isolate them using a number of different methods you mentioned. So we can centrifuge them down to isolate them, and they will remain intact. We can also, because they're bigger, so we can isolate them using the typical cell sorting method, FACS. But once we isolate them, we can actually visualize them variety of different methods. For example, by microscopy, we can see DNA, RNA, and protein of interest, and even whole organelles in there. In terms of whether, because in the area of exosomes and microvesicles, sometimes certain microRNA or certain molecules are enriched in those vesicles. In apoptotic bodies, sometimes we do see them. Again, it's very cell type dependent. So, for example, as I mentioned before, for the monocytes, where they form the protrusion that become beaded, under those settings, nuclear content such as DNA and nuclear protein are excluded, whereas it's mostly cytoplasmic protein in there. But for a number of other cell types, such as T cells, for example, nuclear material can be partitioned. In terms of whether a particular microRNA is enriched in there, it's a bit different because apoptotic bodies, at the end of the day, comes out of dying cells and they get packaged in that setting. So they don't really package, at least in our experience, they don't package a specific microRNA per se. But basically, if that particular cell, before it dies, express, say, high level of a particular microRNA in apoptotic bodies, they're likely to contain the same enrichment.
Oh, okay. So, there's the assumption that it's just, there's no, this cell, I'll just anthropomorphize. The cell has no plan, it just dies. So there's just random stuff in the apoptotic bodies. But what if the death was not only programmed but anticipated? And what if the apoptotic bodies were deliberately packed with some sort of communication, as in regular exosomes? Maybe they're directed at certain targets, and then when they reach the targets, they will influence them in a certain way. Maybe the cell was dying in a way where it says, all right, it's like a will and testament. I want my cellular assets to go to these cells because they need them, or make sure that these go to the to this place to be chewed up and destroyed, and not maybe there's instructions, what I'm saying, on the apoptotic bodies in some sense.
Well, yeah, that's exactly what we are trying to look into. So again, whether the apoptotic cells would then break apart and have certain signals that can actually direct them to a particular cell type is something that we're definitely looking into. But one interesting thing that, under physiological setting, we actually still don't quite understand this process. For example, if a cells die in the thymus, does it go anywhere else other than the thymus, or can it traffic somewhere else? to communicate with other cells. But one thing that we are working on right now is for example, as you mentioned, under certain settings. So, for example, when a cell are, say, infected with influenza virus and then cause them to undergo cell death, apoptotic bodies actually can contain some of those viral molecules, viral RNA, protein, and also virions in that case. And that can be trafficked to neighboring cells. And then based on our data, we think that they can also cause the other cells to become infected. So that's one of the functions. But in terms of targeting them to a specific cell, we still don't know yet. But one thing we do know is that apoptotic bodies contain the surface of the vesicles or apoptotic bodies and also apoptotic cells are changed. So they can expose a particular lipid, such as phosphatidylserine, which can be picked up by certain phagocytes if they have the phosphatidylserine receptor per se. Yeah.
Yeah, what'd be interesting is if you look at a cell that has been, lysed again by a virus or by a virus. If you look at the apoptotic bodies and if you see, a complete absence of the virus in some, or if you see an even amount in each, or it might tell you that perhaps the virus is being able to use the cell's machinery to deliberately make sure that the apoptotic bodies will have viruses in them so they can spread. And if so, if the virus can direct the cell's machinery to do this, that might say that the cell can do this as well. It might have some control over, again, what goes into its apoptotic bodies instead of just randomly blowing open.
Yeah, definitely. In particular, if there's a mechanism where, for example, some virus will assemble on the plasma membrane, so therefore they are naturally they're already at the site where the apoptotic bodies are generated. So I would not be surprised if there's a mechanism where if a cell can actually export certain protein to that particular location or in a traffic to that site, those proteins are more likely to be enriched in apoptotic bodies.
Well, wait a minute. Where do the bodies form? So once a cell has produced them, is there anything left of the cell? Is the center part left and just, the membrane has been budded off completely? Or is it the whole thing gone? The entire cell gets, vesiculated and sent off?
In most of the case, often the whole cell get vesiculated and break off. But more, I guess, for certain cell type, in particular when the vascularization actually happen at the plasma membrane, what happens is often it's the nucleus and part of the membrane that actually get left behind. And often that's also the biggest part of apoptotic bodies and sometimes that part is called apoptotic cell body. So that's kind of like the remnants of things that cannot be, fragment further into smaller pieces.
So what happens to the nucleus, and then what happens to the apoptotic bodies? Like, where do they go? Has anyone traced to see where they end up in the body? Do they all go to the same place or different? Or?
Yeah, very good question. That's exactly what we're working on right now. We do think that, because it contains the nucleus, it probably requires, a particular phagocyte, in particular macrophage, to deal with them. And we are trying to establish methods to actually trying to visualize, does different parts of apoptotic bodies go to different places or being taken care of by different cells. So hopefully in the next year or so, using intravital imaging or also using zebrafish system, that we can actually clearly visualize this process.
Well, what happens, can you find a cell that's getting close to cell death? And if so, perhaps just like in cell division, there's a big rearrangement of the contents, are you able to identify again before it happens, and when it does happen, how fast, and are you able to see what leads up to the happening, the death?
Yeah, I think that the hope is actually setting up, the zebrafish system to visualize this. So if we can, label the plasma membrane, the nucleus, and also other cytoplasmic contents, we can hopefully visualize this, because probably not as organized as a cell division process, but at least we do think that it's not a random process. Say, if we talk about nucleus, we do think that nucleus is most likely to be partitioned. Say a cell would divide it into 10 pieces. The nucleus will probably be partitioned into maybe 1 to 2 or 3 of those fragments. So we're trying to understand, how this process actually occurs.
How early is it for you? What have you discovered about this process so far? And then what are some experiments that you think, what experiments are you conducting that within maybe the next 6 months or a year or 2 years are going to show you something, and what will it be?
Yeah. So we do think that, as I mentioned before, it's a highly regulated process. So we think that during the blabbing stage, and people have described this earlier as well, that, the blabbing force could actually help to push the nucleus to certain part of the cell. And by doing that, and then afterward, the protrusion that we described will then form. And that allows basically the separation of, fragment containing the nucleus and those that don't. So what we're trying to do now is to track whether, does both, say for example, a fragment that contains the nucleus, as you mentioned before, does it stay behind to get cleaved by the local macrophage, and whether the remainder of the smaller fragment doesn't contain the nucleus, actually do play a different role where they can be cleared by other cells, or they can travel further, so therefore they can facilitate an intercellular communication process. Basically trying to dissect, if we do have, if the cells do die and then partition content into different compartments, does any of those vesicles then traffic to different places and play different roles? And that's what we're trying to establish.
Interesting. And what cell type are you focusing on, or cell types? Which ones are the most accessible for you? Which ones die most often?
I think usually it's the immune cells, because there's a lot of cell lines and models that we can use to track them. But we do work on a number of different cell types as well. So right now we know we have a strong interest in monocytes, dendritic cells, T cells. We haven't touched B cells, but we also have a strong interest in endothelial cells as well. Trying to see if they do die, do they just break off into circulation, and after it breaks off into circulation, where do they go? That's what we're trying to work out.
Do cells seem to die one at a time, or what about when a piece of tissue dies? Is there a cascading effect where the dying ones inform the other ones? Is there some signal where you'll have multiple cell die off at once, where it's orchestrated somehow?
There is actually some evidence suggesting that, when cells actually can, release extracellular vesicles to actually communicate, to actually cause neighboring cells to die. In our setting, we haven't looked at that ourselves, so I'm not too sure in our setting. But yeah, something to, worthwhile to look into. I think it will be clear based on some of our in vivo imaging. But so far, when we do the imaging, unless it's during development where a particular area is more prone to cell death, we don't see them, the neighboring cells will die at the same time. Yeah.
Okay, so it seems to be like one at a time.
Yeah.
How long does the process take from, 1 minute the cell's alive, the next minute it's, in a whole bunch of pieces? I mean, how fast does this happen?
It's actually surprisingly quick. So it would take in a matter of minutes. I would say, between 20 minutes to an hour, depending on different cell types again, that they can start from what we can detect them as, cells that are undergoing cell death, have caspase activity, so therefore indicating that cells is undergoing cell death or uptake a particular dye, and then expose phosphorylserine on the surface, so that marks them as dying. And then we can start to see some of those morphological features, like the blebs that I mentioned before, protrusion. And then often within an hour, you will see distinct fragment that actually been generated, and we can capture this by time-lapse microscopy.
What about the microbial attachment, or the viral, the virome, the microbiome, etc.? Do you see any interaction when a cell is undergoing apoptosis with local bacteria or other creatures?
Yeah, so again, depends on the pathogen. Some pathogen would actually prevent cells to die, and some would also promote as well. But in those cases, we haven't quite quantified clearly yet, but we do have feeling that when we do imaging, for example, cells infected with viruses, influenza virus, they tend to break up a little bit better. Say, for example, they generate more apoptotic bodies per cell that actually been influenced by the virus. But sometimes it's difficult to measure that because it takes a longer time, infection, 24 hours until we see the process. So it's hard to capture everything and quantify accurately at this stage.
And what about cancer cells? There's only so many experiments you can do, but have you been able to observe cancer cells in apoptosis, and is it radically different from healthy cells?
Surprisingly not. We, in the lab, I guess most of the time we do, at least in vitro settings, we do use a lot of, cancer cells, and they are very good at dying and then breaking apart when we provide them with a particular stimulus to induce them to undergo cell death. And cancer cells do break apart quite well.
Okay, I just didn't know if they were any different. And then what about cells that turn over quite a bit versus ones that are longer lived? Anyone looked at that to see, like, older cells, are they wiser? Do they break apart differently?
No, good question. We actually haven't really, at this stage, we haven't asked the question, this question of, comparing, if they are, older cells or cancer cells, or in the same cell type, if it's cancerous or non-cancerous, or cells that's more long-lived, do they die differently? I mean, for example, we haven't really even looked at neurons, how they break apart. There's some evidence in the literature when they do break apart, they're a bit different from the process that we are seeing in, say, immune cells, which turn over more. But yeah, we haven't actually looked at that.
Yeah, and then cells in a tissue, if they're in a fixed extracellular matrix.
Mm-hmm.
I don't know if there's a need for them to form all these bodies, and if so, where would they go? Do they, if you're a surface cell but constrained laterally and by extracellular matrix, but if where do your apoptotic bodies go? What if you're an interior cell where you're surrounded on all sides except for a little bit of interstices, by other cells? Like, do you make much smaller bodies so that they can be carried out of there? What happens to you?
Yeah, no, that's another fascinating question. So again, hopefully we can work on that in the zebrafish system. But at least in the preliminary data so far, we don't think that's the key inference. At least I'm pretty sure that will have some inference. But we see epithelial cells, supposedly they get pushed out, and then get removed, but they still form quite a lot of apoptotic bodies, and we wonder why that's the case. On the other hand, cells that are, say, for example, T cells in the thymus, surrounded by, thymic epithelial cells or macrophage, they also break apart extremely well. So, whether there's a, extracellular matrix is going to, or, the local environment causing them to break apart differently is, at this stage, we don't think that would be the case yet.
So what are you hoping to figure out in the near term?
I think the key things that we want to figure out is to try to understand what is the molecular mechanism. So what are the molecules that control this process? But we do have a theory now that we know that plays a key role. But what we do really want to find out is to do with, particular disease settings. Do they actually play a role in particular disease, whether it's infection, autoimmunity, or cancer? Do they actually play a role in communicating with other cells? Because ultimately what we want to do is to identify drugs that can actually either inhibit or enhance how cells break apart. And by doing that, hopefully we can say, for example, if we block cells to break apart during infection, that could be beneficial. Or maybe in a cancer setting, when we enhance this process, that could be beneficial. We don't know.
Well, very good. What's the best way for people to find out more and to get in touch and see your papers?
Yeah, so best to come to the La Trobe website to look for me under La Trobe website. And then from my page, you can see the different themes that we're working on. And then from there, you can find a link to some of my papers.
Excellent. Well, Ivan, I appreciate you coming on. Thank you.
Hey, Chief. Thank you.
You're listening to the Future Tech Health Podcast with Richard Jacobs. Until I reached age 40, I never realized the obvious, that we all have medical issues, or we at least have a family member or close relation that has, or will have them in the future. Medicine and biological systems are the final frontier. Until we've conquered death, figured out how life began, cured cancer, and understood our purpose in the universe, there's a heck of a lot to talk about when it comes to our health. Future Tech Health means I'll be covering futuristic topics that are actually already in clinical trials, or even starting to appear on shelves or by prescription, or available for your own use. We dive deep into stem cells, CRISPR-Cas9, the science of sleep, epigenetics, medical testing, cancer, ketogenic diets, stem cells, aging, regenerative medicine, and more. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a serious medical problem. Remember, however, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you enjoyed the podcast, please listen, subscribe, like, and share it with friends. Thank you.